All right, so hello everyone. Today we are going to have Piotr Sliz, an associate professor in pediatrics at the Boston Children's Hospital and in the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School. He's also a director of the Research Information Technology and Services at the Boston Children's Hospital and an advisor for Research Data Technology Resources and Training at the Harvard Medical School. Piotr is going to be talking about SBGrid, a consortium of over 250 members and several employees supporting its operations. And SBGrid is a tested and refined software infrastructure that includes a large library of scientific applications that has been expanded beyond X-ray crystallography to include electron microscopy and nuclear magnetic resource imaging. So thank you very much for being with us today, Piotr, and the floor is yours.
Thank you, Daniel, and thank you everybody for attending. So I'll just provide a quick overview and feel free to interrupt, ask questions about SBGrid. So effectively, we are an upstream for structural biology, structural science, which includes, as you said, cryo-electron microscopy, NMR, and as well as X-ray crystallography. So all aspects of experimental structure determination. Our team compiles and curates a collection of currently over 1,000 applications. We encapsulate them and make them ready for execution for the members of the consortium. So eventually the applications get installed on the HPC resources, Mac desktop computers, workstations in our member laboratories. Kind of going quickly back the history of SBGrid, we started around 2000, 2001 at Harvard University at 7 Divinity Avenue. Initial scope was to support research groups of Stephen Harrison and Don Wiley, but that only lasted for about 6 months, where we started to, at the time, see the trainees of the research groups as they were leaving local environment and moving up to set up independent positions. They wanted to adopt this similar platform at wherever they were going. So we have Karen Reiners at Yale, Ben Spiller in Nashville, Ishi Tao at Rice University. They became the first members of SBGrid outside of the Harvard community. In additional kind of a significant point was around 2001, 2002, when we joined forces with Tom Walls, who was doing cryo-electron microscopy at Harvard Medical School. At the time, we moved from 7 Divinity to Harvard Medical School, and Tom really collected the enormous collection of cryo-EM images and had this bottleneck, computational bottleneck, just didn't have HPC resources and software available readily to him to process it. We built the first Mosaics cluster, and we expanded our initial collection, which was in 2000, 2001, mostly focused on X-ray crystallography to support cryo-EM. And that has been very productive, and by 2004, 2005, resulted in a number of Nature papers and articles that we have here on the screen, including 2 papers by Alex Fulton, who was a graduate student at the time at Harvard Medical School. So fast forward to today, and that slide I updated just a couple months ago, we are currently supporting just over 500 research groups, I believe 515 as of today, over 160 institutions, 22 countries. And what really started to actually grow during COVID, we also expanded significantly to cover biotech. We have more than 15 biotech members right now within SBGrid. Okay, so that's a little bit history and how we came to where we are today. Just wanted to give you a little bit more information about the tools and resources that we develop and we maintain. So I'm going to start with the structural biology software directory, which is available on our website. So you can go to sbgrid.org/software. And on this page, you see the collection of all the applications that we currently curate with multiple versions and supporting multiple operating systems. So here we can see the count. Over 500 applications are included in this collection. And for each application, we maintain pretty complete metadata on our backend, which includes software description, keywords, links to relevant websites, version numbers, installation notes, and also citations for the particular applications, which is curated by our team and available to our users. We also have a fairly extensive effort to interview software developers, and for many of the applications in our directory, we include extensive interviews with software developers, and we've also recorded over 100 webinars with software developers about the use of individual software applications. And Jason Key, who's here attending today, is leading most of them. So all of this is kind of combined and available through the website. So that's on the structural biology end. We've also more recently, and this really started 2-3 years ago, developed and expanded the separate collection of biomedical software, which is available through this biogrids.org website. And under software subdirectory, you kind of have a very similar structure, listing of all the applications with the similar metadata. I'm not going to go into details about how this is maintained on the back end and our infrastructure, but I wanted to mention that we kind of scaled up what we now call multi-grid environment with capsules set up to support those applications in a very similar fashion to what we do on the structural biology end. Okay, so that's on the website. So once our members can access all this metadata, actually everybody can access metadata on the website. But if you inside the consortium, the next step you can actually use our installation manager and download and run the applications on your local computers. So this is just a quick peek at the installation manager, and it has the look which is in some ways similar to what we have on the website. Users can just go and pick select applications they want to have installed locally. And after the selection is completed, they just click the install button and everything can get installed on local computers. We also have a separate version of this, which can be used by system administrators to install everything at once. And it has a command line interface that will support potentially putting it in a cron job and running it on a regular basis in the background. Okay, so that's the installation manager. Once you have the application installed, then you would move and actually access them locally on the computer, and this is done through the SBGrid shell environment. So once you source our environment, all the applications are available to you for immediate execution. We have a fairly extensive backend infrastructure that ensures that all the applications are compiled and available with all the required libraries, dependencies, etc. So all of that is done for the user. You don't have to modify your .cshrc file anymore after you source our environment. You can have your individual .sbgrid file, which will allow you, for example, to select a specific version of the application. Within our collection, some application might have as many as 10 different versions. And we actually, on the back end, we maintain all the versions going back to 2005 or so. Some of them we pull out of the distribution, but if somebody wants to reproduce particular workflow that they've done, let's say 10 years ago, we can easily bring back this application and make it available to our user community again. Okay, so that's the shell. So that's effectively all about the execution environment, and we just recently submitted the paper that in details goes into the capsule environment that Jason and Ben developed within SBGrid. We can answer more questions about it. But I wanted to kind of quickly deflect to our Upsider citation tool. And this was developed by Stephanie Socia. So, that's her picture here. And within the tool, when you go to our software directory, for every single application, you can effectively click the button and kind of very rapidly build up collection of the citations for all the applications that you use in your particular project. And we feel this was in particular quite important for our community and for the developers community, who often is not fully recognized as the publications which fully depend on the application's software that was being used being published. So this is embedded within our infrastructure. Another collection I mentioned is educational videos, and with software developers created by Jason and others are available on the YouTube channel, and this is available to everybody. So you can go to youtube.com, SBGrid TV, and access this collection directly. Last few minutes of the presentation, I wanted to dive into SBGrid Data Bank. And this is the project that has been completed in collaboration with Harvard Dataverse, with Merce Crosas, and you can see her name here on the publication from Nature Communication in 2016. And Peter Meyer, who is here on the call as well, was the first author on the paper. And we've used initially our own database, eventually merged it with the Dataverse course and extended Dataverse to support the structural biology collection. And what we did, we created this repository of over 750 structural biology data sets, now contributed by over 100 individual research groups. And within this repository, you can have nice viewers. You can look at the structures, which corresponds to the data sets. We mint the digital object identifiers, so all the data sets could be then subsequently referenced in publications. And we also developed this highly replicated high-performance computing storage capacity, which makes the data sets in the collection immediately available for reuse and potential computations on the HPC resources. And Pete also developed this on-the-fly processing pipeline. For every data set which is deposited in the collection, we automatically try to validate it. And here you can see 4 different pipelines that actually run through the data sets, and all of them completed successfully. And this has been quite important because it immediately kind of confirms that the data set, we have all the metadata to potentially reproduce and the results from the corresponding publication. Okay, and I mean, the last bit here, Pete also created the training collection. So we have this set of 12 data sets, which really present a diverse set of challenges in the structure determination by X-ray crystallography. And we encourage people to use it as a training set as they're working on their own data. If they want to become really fully proficient with X-ray crystallography, they can go and process those 10 or 12 data sets to completion, and this will like really fully make you competent with varieties of structure determination approaches. Okay, so maybe I'll finish with that. I don't know, we've been like 8-9 minutes. I can go on about the use of SBGrid in the local community, and in particular with Ben on the call today. One of the cool things we've done over the last year was redeploy the SBGrid platform in AWS cloud environment. And the idea here is to make it kind of on-demand and rapidly available to the communities that might not necessarily have local computing resources to support their research. And Ben is here if you guys have any questions, we can take it on. Okay.
Thank you very much, Piotr. All right, so I'd like to open the floor for questions.
I have a question. I'm very interested in the AppCider, in the software citation thing. Where do you get the citation information? Do you create the citation from the SBGrid or BioGrid resource itself? In other words, are you asking for citations to BioGrid itself for use of the software?
No, so the citations is mostly manual process. So we include an application from a developer in UK. We would go and look at their website, and if they have this information, we would put in our database. If this information is not available, we might email them directly and ask them. So it's kind of a manual research process to find the best sort of citations for any particular software title in our collection. And also, if we get it wrong, the developers are not shy about emailing us and correcting us or what needs to be cited. And sometimes they proactively, if they have the new paper, they will reach out to us and tell us there is a new publication. Can you please change your information?
Okay, so
one I guess possible point of clarification there is that AppCider is a tool for finding manuscripts that the developer has written to cite for their software versus a citation to the software itself.
Okay, I think I need to maybe look at this to understand what's going on. So I come from astronomy, and astronomers generally have a way that they want the software cited. Sometimes it is a paper in which they describe the software. The best practice generally for software citation is becoming increasingly that you want to cite the software directly, independent of a paper, although you can also cite a paper. So there are different ways that one can cite the software directly, such as the ASCL entry, a Zenodo entry, some other thing, or the paper, etc. So I'm
So
wondering.
in our field, Alice, is mostly the paper that they publish about the software, and
Okay.
because how the funding works, they really need to in structural biology at least, right? Almost every title in our collection would have the corresponding publication when they described how the paper with this data citation, we really meant the citation, which includes DOI. So we create a new citation, which is just the data citation. So we do it using our kind of programmatic approach, but for almost all the software, we really find the paper. We haven't really kind of created new citations directly to the software title, if you wish.
Okay.
Does it make sense? Or?
Yeah. So this app citer then has the information about the papers themselves. I'm trying to understand. You're using
so it's basically a database. In our internal database, we have a software title, and then we would have the paper that's cited that describe that software. So it's our internal database of software and the citations by the authors of the software. And then we just basically, then you select the titles, and then we create an EndNote library for you that has the 20 or so papers that you should cite as you're writing the paper. So we just give you an EndNote library at the end.
Okay, for all those different software packages that they made.
For the 10 titles that, let's say, you use for your project.
Okay, great, thank you. That's interesting. I'd like to kind of look at that maybe.
Yeah, it's a lot of manual work. So we have like literally, Jason, 3 or 4 people working on it between Michelle, Satara, curator, right? It's each title. But once we have them, like they don't change that much. So a lot of work is when we onboard a new application to make sure that we have the right citation.
Yeah, so we list it. ASCL has a preferred citation field where we list how the software author wants the software cited. And that can be like all kinds of different crazy ways. Sometimes it's just a link to look here because the instructions are so complex. But okay, good. Thank you.
And sometimes, especially for older titles, the challenge is to kind of really engage in that communication with the author of the software. Like they not always reply, right? So sometimes it works, sometimes it doesn't, right? In particular for us, we have a lot of older applications as well. It's not kind of a new front of the open source code that comes out and people are on it, all the time.
Yeah, I just add that it's also kind of dynamic because the ease at which labs and scientists have been able to write and distribute software via code repositories like GitHub and publish papers immediately in like archive and bioRxiv have made it easy to get code out, get an unreviewed manuscript out, throw that out as the citation, but then you have to follow up, right? Because that bioRxiv, those are just papers that have been submitted in other places. Sometimes they get rejected, sometimes they never get published. Sometimes it takes a year as they bounce around. So we have a team who sort of help us track those things, but it's a dynamic and kind of ongoing process.
But it's not uniform. It's very, because we have some application, the author would like you to cite 10 papers when you use their tool, right? And then there are limits how much you can cite. So we have, it's a variety of scenarios, right? We would export everything. So if somebody wants us to cite 10 papers, we'll create EndNote with those 10 citations. Ultimately, whoever will use it for publication, they need to decide what's the right balance.
Great, thank you so much.
We have time for one more question. If no one has one, then I'll have one. So you mentioned the amount of effort that goes into curating citations. I was wondering the amount of effort that goes into curating the rest of the metadata associated with each entry. Can you elaborate a
So
little
maybe,
bit more on this process?
curating software itself, when we compile and build and then put it all together, we have, I would say 4.5 full-time staff right now. And within the curation process, this metadata it's often part of the intake, right? When you take the new version of application, try to compile it, you take the data, we put it into a database, but we have also administrative stuff that will help to make sure that this is complete, right? So in terms of people, like, everybody on the team, but the metadata, I mean, it's hard to kind of quantify how many minutes, per title.
And I was wondering, also how much is the author involved in this reproducibility effort? Like, do they have? I was wondering, like, if I want to submit a new software component and you want to make sure that it's part of the reproducible pipeline, is that something that you guys would do, or is the author supposed to follow a process to ensure that?
We mostly grab it from the websites and different places when people put it out in our community. People would often have it available on the website, so it's mostly from our end. Although we have number of developers that would email us or give us like, this is the latest version, right? But definitely, if anybody kind of approaches us, can you include my software? I think this will be applicable to our community. Jason, you guys would take it, right? I mean, you can comment more here about
Yeah, I
the
think
balance.
we apply sort of a first pass filter. Is this domain appropriate? Like, is this a software title that would be useful in structural biology or bioinformatics? Then is this something we can support? Some software is, it's pretty tough to support, and there are architectural considerations that if the software developer decides to implement a database or some sort of difficult to support in a read-only software stack, that, obviously anything that requires a web interface or accounts or something like that gets challenging. If it meets those requirements, once we take it, we're sort of committed to supporting it and maintaining it for the operating systems we support. We've had everything from, we have software that's kind of aged out, right? That, was written on like Red Hat 5 or RHEL 5, and where we went to 6, we went to 7, we would at some point it's going to stop working. Some of those packages, we benefit from the fact that older software tended to be better written than the sort of newer software that we're dealing with. But so those are easier to work with. But once the underlying operating system goes end of life, then we may have to start to rely on things like containers and such to implement that. But luckily, the tools are started keeping pace. So far, we don't have anything that we haven't been able to.
And Daniel, if you
resurrect.
have interesting title we would think will be applicable, we can kind of do like a little campaign to make it. We put in a newsletter on the website. We might do a webinar with you. Just let people know that this became available, right? We can also track the usage, so we can see how quickly applications are disseminating, the use is disseminating through the community, so we can kind of use it to help us. And I'll ask how many people. I mean, altogether the team is around 15 people right now. This is between the curation and then just kind of building innovation, the new things we're building, administration, and everything else. It's around 15 people.
Thank you.
All right, I think we have run past the time that we have for questions. So if you have I'm gonna stop the recording right now. Thank you very much, Piotr and team.